Okay, those joining by Zoom, okay, it sounds like we do have audio now. Mute this. Okay, so for those joining by Zoom, I am doing something a little bit different today. I noticed that as I'm pacing around the room, my voice kind of goes in and out as I walk away from the microphone. So I'm trying a different microphone that I'm wearing today. Hopefully this will work a little bit better. Okay, so before we get started, a couple quick announcements. Quizzes, so we'll start quizzes up again today. Quiz number 7 will be today. That'll be on the material that we covered in Wednesday's lecture and today's lecture. As far as the exam goes, I haven't looked at the class average yet, but I saw a lot of good scores. You guys did pretty good. Remember that I made a mistake when I gave you guys the whole period. I usually do that for written exams, but when it's online, I usually give you guys about 45 seconds per question. So remember, the next test, you're going to have a little less. You're going to have about a half hour to complete it, which is no problem for most students. Also, the test, students who missed the test haven't gotten back to me about makeups yet. So I'm just going to go ahead and set up the exams so that you can look at your answers, see what you got right and wrong, so that you can study for later on. Now, your old exams are going to be very important later on in the semester because our final in this course is a cumulative or a comprehensive final rather. So that means it's going to be on everything that we've covered the entire semester. This makes your old exams a really valuable resource for studying. Okay, the best study guide you guys are going to have for the final are your old exams. Okay, so I'll make the exam available, and at least try to access it so that you know when it comes time to study for the final that you'll be able to have that available to you. Okay. And I think that's about it. There was a question on Wednesday before the end of the class, a little confusion about what group is meeting today. I forgot to look at the calendar and send an email to you guys. However, Group A was supposed to meet today. If anybody's here who's not in Group A, that's okay. Just sign the attendance sheet in case we have to do contact tracing. But yeah, remember, so going forward after exam number 1, today is Group A's Friday. Next Friday will be Group B's Friday. And I think that's about it. Any questions on any of that? Okay, if there's no questions, let's get back to Chapter 4. So we should be able to finish Chapter 4 today. This morning, I just posted the Chapter 5 PowerPoint. I'm not sure if we're going to get to that. Looks like probably not, since I already wasted 5 minutes messing with the computer and making announcements. Okay. So when we left off, we were talking about cell membranes. Okay. Now, of course, cell membranes are very important because they're a barrier between the extracellular environment and the intracellular environment. They provide a distinct intracellular space, and they can control the passage of molecules from the extracellular space to the intracellular space and vice versa. So they're very important. We know that a major constituent of cell membranes are phospholipids, specifically in a phospholipid bilayer. Now, of course, we talked about the fluid mosaic model on Wednesday, so we know that these phospholipid bilayers of cell membranes in particular are fluid structures, meaning that the proteins, the sterols, the phospholipids, the glycolipids that make them up are free to drift around and rotate. We also know that it's so the second part of the fluid mosaic model states that a membrane is a mosaic or a mixture of different things. Okay, so we've got phospholipids making the phospholipid bilayer. We've got sterols. Okay, cholesterol is a component of a cell membrane. We've got proteins. Okay, we've got lots of membrane proteins, transport proteins, receptor proteins, cell recognition, all sorts of different proteins with different functions. And we've got glycolipids as well. Now, in the absence of all that other stuff, if we just have a plain membrane made just of a phospholipid bilayer, it is selectively permeable. Okay, what selectively permeable means? This means that some things can pass through it and other things cannot. Things that can pass through it include small hydrophobic molecules. Okay, so small nonpolar molecules, things like O2, molecular oxygen, CO2, carbon dioxide, N2, molecular nitrogen, benzene, pardon me. All of these things, excuse me again, all of these things can pass easily across very freely across a phospholipid bilayer. Small uncharged polar molecules like water, glycerol, ethanol, pardon me, I don't know what's wrong today. These can cross the phospholipid bilayer, but not freely. All right, they need something pushing them. They need we'll talk about osmosis a little bit. They need some osmotic pressure in order to cross that phospholipid bilayer. Okay, so they can cross, but they can't cross easily like small nonpolar hydrophobic molecules can. Larger uncharged polar molecules, so if the molecule is too big, it can't pass. Things like amino acids, glucose, nucleotides, and ions, even though they're very small in size, anything with a charge cannot get past this hydrophobic barrier that the fatty acid tails of the phospholipids make. Okay, so selectively permeable. Some things can pass through, some things can't. This week's lab, good example of selective permeability, right? Most of you should have had it done by now. So rather than a phospholipid bilayer, you're using a dialysis membrane that has very small microscopic pores in it. Okay, these pores are large enough to allow water molecules through, but too small to allow sucrose molecules through. Okay, so again, selectively permeable. In the case of the dialysis membranes you're using in lab this week, selectively permeable based on size. Okay, in the case of a phospholipid bilayer, selectively permeable based on hydrophobicity and size as well, small hydrophobic molecules. Okay, diffusion. This is another concept that you probably talked about in lab this week, or will talk about if you have lab today. Heat energy causes the random motion of molecules, and at a higher temperature, we have more motion. Okay, so usually in class, I'll use a cup of coffee as an example, but of course with the pandemic, we're not bringing our coffee to class these days. But if you imagine in a cup of coffee, the water molecules in that cup of coffee are in constant motion. If it's a hot cup of coffee, those water molecules are moving faster and colliding more. So heat energy causes the random motion of molecules. For example, the water molecules in a cup of coffee. And the higher the temperature, the faster the random motion of those molecules. Brownian motion is the random movement of small particles resulting from the impact of moving molecules. Okay. So again, this is something that most of you have probably already seen in lab this week, except for those who have lab today. One of the exercises in lab this week is to look at India ink suspension underneath a microscope. You know, India ink is actually a suspension of very tiny black particles. And if you look very closely at the black particles, they're kind of vibrating and moving randomly. The reason for this is because These India ink particles are in a suspension with water, and the water molecules are in constant motion. And the collision of the water molecules with these small particles is what causes them to vibrate randomly and move randomly. This is Brownian motion. Brownian motion, the random movement of small particles resulting from the impact of moving molecules. Okay, so hopefully everybody had a chance to see that in lab this week. On a small scale, Brownian motion appears to be random. However, on a larger scale, it's not random. There's a net movement of molecules in one direction. So looking at the last bullet point here, the net movement of molecules as a result of energy is not random. In the absence of other forces, a substance over time always moves down its concentration gradient. Okay? Specifically from an area of high concentration to an area of lower concentration. So I've queued up a YouTube video for you to demonstrate diffusion. Hopefully this is the right one. We'll see. Okay, I think this is the one. Let's have a look. Great. We can't escape from ads in class. There we go. Okay, so what he's doing, he's putting some ink in water. So the drop of ink, once he puts it in the water, that's an area of high concentration. Now, the water is going to move down its concentration. Notice there's cold and hot water. Of course, the hot water, we're going to see diffusion happen faster because everything's in motion faster. There's the cold water. We can see the drops are the high concentration. Now, eventually, it'll move to areas of lower concentration and be a homogeneous color. You can see how the ink is diffusing faster in the hot beaker of water. Again, going from an area of high concentration to lower concentration. Here's an area of high concentration. Over time, that will diffuse to an area of lower concentration. Okay, the vinegar, that's some kind of chemical trick he's doing, but all we have to worry about right now is the diffusion. I think this is actually the, yeah, let's give this video a look. I think this one's a little bit better. Oh, enough with the ads. Let's see, how long is this? Oh, we're not going to watch all 5 minutes. I'm going to skip ahead. Okay. Hot water, cold water. Okay, so we can see areas of high concentration of ink in the water, and over time, the ink molecules are going to move from the areas of high concentration to areas of low concentration until we have one homogeneous color in each of these. Okay, now it's going to happen faster in the hot glass of water than in the cold glass of water, but over time, eventually, the glasses are going to be one even homogeneous pink color. Okay, and we can see how it's going a little bit faster in the warm water than the cold water. Okay, that's enough. I will post links to these on eCampus if you want to look at them later. Okay. Oh, I'm sorry, I've been neglecting the Zoomers. So I see the Zoomers appreciated the video. That's good. You're welcome, guys. So again, on a small scale, we have Brownian motion, which appears to be a random movement of particles due to collision with molecules. However, over a larger scale, the movement isn't random. We go specifically from an area of high concentration to an area of lower concentration. Whoops, I'm going the wrong way. There we go. Okay, so some definitions. A concentration gradient is a difference in the number of molecules or ions per unit volume between adjoining regions. Diffusion is the passive movement of molecules or ions down a concentration gradient. And again, but when I say down a concentration gradient, I mean from an area of high concentration to an area of lower concentration. And osmosis is diffusion of water across a membrane down its concentration gradient. Okay. So again, this week it seems like lab got a little bit ahead of lecture. If you had lab already this week, you probably learned a little bit about osmosis already. But what osmosis is just diffusion under a certain set of conditions. Okay, so diffusion of water across a membrane down its concentration gradient. So let's talk about osmosis a little bit. Looks like everybody's almost done with this slide. Okay, so again, you guys, hopefully you guys already were exposed to osmosis through lab this week if you had lab. But let's consider this slide. So we have a receptacle here that's split in two by a semi-permeable membrane. Okay, so on the left, we have just plain water. On the right-hand side, we have a mixture of protein and water. In between these two sides, we have a semi-permeable membrane. Okay, in this case, the semi-permeable membrane is semi-permeable based on the size of the molecule. So you can see we have pores. Let me write on. Okay. So notice that we have pores here, here, so here, where these arrows are. And notice that these pores are large enough to allow water molecules through, but too small to allow protein molecules through. Okay? So over here, we have 100% water. And over here, we have a 20% solution of protein. Okay? If we have a 20% solution of protein, what's the percentage of water then? 80%. Okay? So now, knowing that substances diffuse down their concentration gradient from an area of higher concentration to lower concentration, where's the area of higher concentration in this figure, on the right or the left? So we have 100% versus 80%. Which higher concentration, 100 or 80%? 100%. Okay, so we have high concentration of water here, lower concentration of water here. So knowing that diffusion goes down the concentration gradient, would we expect a net movement of water from the left to the right or from the right to the left? From the left to the right, exactly. Okay, down its concentration gradient. Let me draw on. So we have high concentration here, 100%, low concentration here. So we would expect a net movement of water molecules from the tank Well, the side of the tank on the left to the side of the tank on the right. Okay, protein molecules are going to stay over here on the right-hand side because the pores are too small. Okay, this is semi-permeable. Pardon me. The semi-permeable membrane is selective by size. Protein molecules are too big to pass through. All right. So look at the level of the water on this side and the level of the water on this side. What do you think is going to happen if we let this go for a while and we get net movement of water molecules from the left to the right? How do you think that's going to affect the levels of water on either side of this receptacle? So over here on the 100% side, is the water level going to go down or up? It's going to go down over here, okay, because we have a net movement of water molecules over here. And yeah, just like I see somebody else chimed in on Zoom, and on the right-hand side, the water level is going to go up, okay? All right, let me make sure I have, you guys can see the whiteboard. Now, there are some terms that we use to describe concentration gradients, areas of low concentration and areas of high concentration when we're doing osmosis or diffusion. All right, let me just erase this. Okay, for those of you on Zoom, you're just going to see like a blank document. I'm sharing this blank document just so I have a nice whiteboard to draw on. So pay attention to the camera rather than the screen I'm sharing. So like I said before, there are some terms we use to describe areas of low concentration, areas of high concentration, and areas of equal concentration. These terms are hypertonic, hypotonic, and isotonic. Okay, and these are relative terms, which means they only make sense when we're comparing them to something else. The word hypertonic means having more solute than. Hypotonic means having less solute than. And isotonic means having an equal amount of solute. Okay, hypertonic having more solute than, hypotonic having less solute than, isotonic having equal amounts of solute. So again, for example, in one of your lab exercises this week, you're going to be observing osmosis in containers with different concentrations of sucrose. No, that's not true. You're going to be doing, there's one concentration of sucrose in the beaker. There's 3 different concentrations in your dialysis bags. So here's your beaker that's filled with solution, and you have 3 dialysis bags in there. One with plain water, one with 10% sucrose, and your last dialysis bag has 20% sucrose. Okay. And the solution in the beaker is 10% sucrose. Okay, so let's apply these terms to the experiment that you're doing in the lab this week. So the plain water that's inside this first bag, is that hypertonic, hypotonic, or isotonic to the 10% sucrose in the beaker? Hypotonic, okay? 10% sucrose here, no sucrose here, more solute in the beaker. Okay, so this is hypotonic to the solution in the beaker. I saw somebody. Angle camera down more, please. Sure. Better, I hope. There we go. Okay, let's do 10%. So we have 10% sucrose in this dialysis bag, 10% sucrose in the beaker. So which term would we use for this? Is the solution in the dialysis bag hypertonic, hypotonic, or isotonic to what's in the beaker? Isotonic. 10% sucrose here, 10% sucrose here. Okay, that's equal amounts of solute. All right. Let's do the last one. 20% sucrose in the bag, 10% sucrose in the beaker. So the solution in the bag, hypertonic, hypotonic, or isotonic? Hypertonic, yes, hypertonic. Okay, and remember that these are relative terms. So if we compare the solution in the 20% bag to the solution in the plain water bag, is this hypertonic, hypotonic, or isotonic to that one? 20% versus 0%? Hypertonic, right? Hypertonic. Okay, so I think you guys understand that. We spent enough time on it. All right, let's go back to the PowerPoint. Okay. So how do molecules cross membranes? I've mentioned several times now that cell membranes are barriers that control the passage of molecules into and out of the cell. So how do molecules cross membranes? First way is by direct diffusion. So remember, if we have small nonpolar uncharged molecules like O2, like carbon dioxide, like CO2, like N2, these can diffuse directly across a cell membrane without any energy required. Okay, again, but remember that diffusion only happens down the concentration gradient. Okay, so as long as there's a concentration gradient of small nonpolar uncharged molecules, they can move across the plasma membrane freely, the cell membrane. Another way is facilitated diffusion or passive transport. So if we have molecules that are polar or charged and we need to get them across the cell membrane, well, they can't cross the cell membrane by direct diffusion because they have a charge or they're too large. Okay? However, there are proteins that form pores in the cell membrane. So by facilitated diffusion, again, anything that's charged or polar can cross the cell membrane by going through the pore, but again, it's going down its concentration gradient. Okay, we're going from an area of high concentration to an area of lower concentration. Number 3, we have active transport. Active transport of substances through pore-forming proteins against the concentration gradient. Okay, so in this case, we're taking something from a low concentration and moving it to a higher concentration. Now that's going against the concentration gradient. Okay, that's the opposite way from what the diffusion works. So we need energy. Okay, if we're going to move molecules against the concentration gradient, we need some sort of energy. Okay, that energy can come from ATP, that energy can come from sunlight, or it can also it can come from indirect active transport, which we'll talk about in a little bit as well. Okay, so the 3 ways that molecules can cross membranes: direct diffusion. Again, this only applies to small nonpolar uncharged molecules like oxygen and CO2. They just directly go down the concentration gradient across the membrane. Facilitated diffusion, for anything that's charged or polar, it needs to go through a pore-forming protein, again, down its concentration gradient. If we need to move something that's polar or charged or too large against this concentration gradient, we're going to do that by active transport. This requires an input of energy. So let's look at some examples. I'm sorry, there's a 4th type. Bulk transport into a vesicle. Bulk transport. So there's 3 types of bulk transport. Exocytosis is bulk transport of molecules in a vesicle to the outside of the cell. Right. So you guys might remember earlier in chapter 4, I talked about the secretory pathway. So in the secretory pathway, the transport vesicle goes from the ER to the Golgi, then the Golgi to the plasma membrane. When that vesicle fuses with the plasma membrane and secretes the proteins to the outside, that's an example of exocytosis. Okay, it's bulk transport of those proteins to the outside of the cell in a vesicle. We can think of, so endocytosis is just exocytosis in reverse. So if you can imagine, instead of a vesicle fusing with the plasma membrane and depositing its contents outside, if you can imagine a little indentation forming in the plasma membrane that eventually pinches off into a vesicle and takes substances inside the cell with it, that's endocytosis. Okay, just the opposite of what happens in exocytosis. Then we have phagocytosis. Phagocytosis is endocytosis of whole organisms or fragments of organisms. So last week you had an opportunity to look at amoebas. The way amoebas eat, if you're really lucky, you got a chance to see one of these eating. You probably didn't. But the way they eat is by engulfing entire organisms, and then they take that organism inside of the cell in a vesicle. Okay? Certain cells like amoeba use this to eat. There are also cells of our immune system that are constantly patrolling our tissues for any foreign invaders. And if they come across foreign invaders, they'll do the same thing. They'll do phagocytosis. They will engulf it and then digest it in order to neutralize the threat to the body. All right. Let me just see, before I continue, there's an animation I have on my computer. Last time I tried to access it, I think the file was corrupted. Let me see if it'll work for me today. There it is. Okay, this was a really good example of how exocytosis works. It looks like well, maybe not. No, it's working for me. Okay, I'm going to share a video from my laptop so you guys can see this real quick. Okay, so here's this short animation. I'll post this in eCampus for you guys as well. So this is an example of exocytosis. So you can imagine that these blue dots inside the vesicle are proteins going through the secretory pathway. So the vesicle, you can imagine this vesicle is coming from the Golgi to the plasma membrane. It fuses with the plasma membrane and then secretes the proteins to the outside of the cell. That's exocytosis. If you can imagine, if I could run this animation in reverse, and instead of fusion, we have a budding off of a vesicle from the plasma membrane that contains stuff from the outside, that would be endocytosis. Okay, phagocytosis is the same as endocytosis, but on a bigger scale. I actually have a nice animation in the PowerPoint that demonstrates phagocytosis. Okay, I'm going to switch back to the other computer. Okay. So here are some examples. On the far left-hand side, let me get my little, okay, so over here we're looking at direct diffusion across the plasma membrane. So if we have in black here, if we have small nonpolar uncharged molecules like oxygen or carbon dioxide, they don't need any special proteins or anything. They can diffuse directly across the plasma membrane. But again, from an area of high concentration here to an area of lower concentration. Okay? So going from an area of high concentration to lower concentration doesn't require any energy at all. Okay, so this can just happen spontaneously. Over here, these are two examples of facilitated diffusion. So facilitated diffusion, so now we have some molecules, maybe these are charged or polar. They can't cross the plasma membrane because they're charged or polar. So they need to go down a pore protein in the plasma membrane. So again, they go down from an area of high concentration to an area of lower concentration, down their concentration gradient. All right, so again, going down the concentration gradient, no energy is required. All right, and we're just looking at two types here. This type, some of these pore proteins are gated. So you see this little ball here? So this can block up the pore. So this can either be open or closed depending on the conditions that the cell needs to meet. Okay, so again, this is another example of passive transport or facilitated diffusion. Active transport, so now look, we're going from an area of low concentration to an area of high concentration. In order to do that, we need to put energy in. Okay, we need to hydrolyze ATP. We need to use energy from sunlight, or we need to couple this to another concentration gradient. That's another way to do it. But we need some source of energy to do active transport, which is moving against the concentration gradient from an area of low concentration here to an area of high concentration here. Okay, down below, so I showed you that nice animation of exocytosis. So we get fusion and then release to the outside of the cell. Again, endocytosis is just the same thing in reverse. So here we have a little, this is called an invagination of the cell membrane. So this invagination of the cell membrane gets deeper, pinches off into a vesicle so that we take stuff from the outside of the cell into the inside of the cell. Oh, let me erase that stuff real quick. Where's my eraser? There it is. Okay, then we have phagocytosis. So I have this animation to demonstrate phagocytosis. There we go. Okay, so what we're looking at, the cell that we're looking at in the upper right of this animated GIF file here, let me erase that stuff first. Okay, so this, hopefully everybody can see my cursor. This bigger cell here, sort of in the upper right, this is a mouse macrophage. Macrophages are cells of your immune system that patrol your tissues looking for anything out of the ordinary so that they could stop an infection before it begins, basically. So this is a macrophage from a mouse. This is a fungal spore here. So what's happening is this macrophage, the cell of the mouse's immune system, is recognizing this fungal spore as something foreign, something that's not supposed to be there. So it engulfs it by phagocytosis, and later this mouse macrophage will digest that fungal spore so that any threat to the mouse's health is then neutralized. Okay, so that's an example of phagocytosis. Again, phagocytosis is bulk transport of entire organisms or parts of organisms into the cell. Okay, so types of membrane proteins. Actually, I'm looking at the clock and we're out of time. We'll finish up these last two slides on Monday. Then on Monday, we'll start chapter 5. Again, let me remind you, quiz number 8, I believe it is, will be available online by about noon today. Also, look for an announcement on eCampus. I'll make your tests available to you so you can look them over, see exactly what you got right and wrong. I think that's about it as far as announcements go. Everybody have a nice weekend. If there's no more questions from those Zooming in today, I'm going to stop the meeting, and I'll see you guys on Monday.